Human behavior genetics is a subfield of the field of behavior genetics that studies the role of genetic and environmental influences on human behavior. Classically, human behavioral geneticists have studied the inheritance of behavioral traits. The field was originally focused on testing whether genetic influences were important in human behavior. It has evolved to address more complex questions such as, how important are genetic and/or environmental influences on various human behavioral traits? To what extent do the same genetic and/or environmental influences impact the overlap between human behavioral traits? How do genetic and/or environmental influences on behavior change across development, and what environmental factors moderate the importance of genetic effects on human behavior? The field is interdisciplinary and draws from genetics, psychology, and statistics. Most recently, the field has moved into the area of statistical genetics, with many behavioral geneticists also involved in efforts to identify the specific genes involved in human behavior, and to understand how the effects associated with these genes changes across time and in conjunction with the environment. History. In 1869, Francis Galton published the first empirical work in human behavioral genetics, Hereditary Genius. Here, Galton intended to demonstrate that a man's natural abilities are derived by inheritance, under exactly the same limitations as of the form and physical features of the whole organic world. Like most seminal work, he overstated his conclusions. His was a family study on the inheritance of giftedness and talent. Galton was aware that resemblance among familial relatives can be a function of both shared inheritance and shared environments. Contemporary human behavioral quantitative genetics studies special populations such as twins and adoptees. The initial impetus behind this research was to demonstrate that there were indeed genetic influences on human behavior. In psychology, this phase lasted for the first half of the 20th century largely because of the overwhelming influence of behaviorism in the field. Later behavioral genetic research focused on quantitative methods. Contemporary behavioral quantitative genetics. Behavioral geneticists study both psychiatric and mental disorders, such as schizophrenia, bipolar disorder, and alcoholism, as well as behavioral and social characteristics, such as personality and social attitudes. Recent trends in behavior genetics have indicated an additional focus toward researching the inheritance of human characteristics typically studied in developmental psychology. For instance, a major focus in developmental psychology has been to characterize the influence of parenting styles on children. However, in most studies, genes are a confounding variable, because children share half of their alleles with each parent. Any observed effects of parenting styles could be effects of having many of the same alleles as a parent. Thus, behavior genetics research is currently undertaking to distinguish the effects of the family environment from the effects of genes. This branch of behavior genetics research is becoming more closely associated with mainstream developmental psychology and the subfield of developmental psychopathology as it shifts its focus to the heritability of such factors as emotional self-control, attachment, social functioning, aggressiveness, etc. Several academic bodies exist to support behavior genetic research, including the International Behavioral and Neural Genetics Society, Behavior Genetics Association, the International Society of Psychiatric Genetics, and the International Society for Twin Studies. Behavior genetic work features prominently in several more general societies, for instance the International Behavioral Neuroscience Society. Methods of human behavioral genetics. Human behavioral geneticists use several designs to answer questions about the the nature and mechanisms of genetic influences on behavior. All of these designs are unified by being based around human relationships which disentangle genetic and environmental relatedness. So, for instance, some researchers study adopted twins, the adoption study. In this case, the adoption disentangles the genetic relatedness of the twins from the family environments. Likewise, a classic twin study contrasts the differences between identical twins and fraternal twins within a family compared to differences observed between families. This core design can be extended, the so-called extended twin study, which adds additional family members, increasing power and allowing new genetic and environmental relationships to be studied. Excellent examples of this model are the Virginia 20,000 and the QIMR twin studies. Also possible are the children of twins design.